Okay, hi there everybody. My name's Emily Perry. I hope you're all here for the Week Resources in Ensembl Plants webinar. So what I want to talk about to start with is why we need genome browsers. Well, we started out in 1977 with 5 kilobases of a bacteriophage, which was sequenced by Fred Sanger. In 2004, we finished, very much in inverted commas, the human genome sequence, which was 3 gigabases. And now we're looking at things like the wheat genome, which is 10 times that. And the data that we get when we start with is just this. It's A's, T's, G's, and C's. It's not meaningful by itself. And genome browsers are a way of turning this kind of data into something that's human readable. So Ensembl Plants is a genome browser. We have loads and loads of data. So we have genomes and annotation for 58 plant species. We compare all the genes that we've produced on these genomes. We compare them to each other, and we produce gene trees. We have ways that you can display variation data, and we also have a tool called the Variant Effect Predictor, or VEP. We let you display your own data. We have a tool for data export called Biomart, and we also allow programmatic access. We have a Perl API and REST API, and we are completely open source. You can access the data at various different scales. So if you're interested in single genes or single variants, you would probably access the browser at plants.ensembl.org. If you're looking at groups of data, you'd most likely be looking at Biomart for downloading tables of data, or our REST API, or maybe the Variant Effect Predictor to find out about lists of variants. If you were looking at the whole genome in one go, you would use our FTP site to download things like whole genome FASTA files or GTFs of the annotation, or you could potentially use the Perl API or the MySQL database to access at any scale you like. We release our data on a release cycle, so every 2 to 3 months we will put out a new release. So we're currently on release 42, which came out just last month. And in the course of the months following, we will bring in things like new genome assemblies, updates to underlying software, updates to gene set. If we've got new genes and genomes, we have to compare them again. Updates to variation data in any new or updated interfaces, and we plan to bring the next one, Ensembl 43, out in April. That should say 2019, but it says 2018. Made an error there. So we're here to talk about wheat, and wheat is obviously a very important genome. So wheat is an important crop plant. It's grown on more land area than any other crop at all. It's cultivated all over the world, in every continent except for Antarctica, for obvious reasons. And there are more varieties of wheat than there are for any other cereal crop. So you can get about 5,000 cultivars of bread wheat that are in current use. And it's being harvested around the world somewhere every month. And it's important in developing countries. So there's 1.2 billion people who depend on wheat for sustenance and for their livelihoods. And the demand is projected to increase. So with the population increase in the developing world, it's thought that this will increase by 60% in 2050. But the genomic revolution gives us hope. We can improve yield under different conditions. We can, to be a little bit over the top, we can feed the world. Wheat, however, is not easy. And this is why. So wheat has a hexaploid genome. It is not an easy genome to work with by any stretch of the imagination. So it started out as 2 different genomes in, and I'm sorry if I mangle the Latin here, Triticum auratu and Aegilops splatoides, what are known as the AA and the BB genomes. These hybridized to produce Triticum dicotyloides, AABB, which then hybridized with Aegilops tauchi, that was terrible Latin, the D genome, and we ended up with Triticum aestivum, which is our current wheat genome, which has got 6 copies of each chromosome, because of course these were all closely related species that underwent this hybridization. So we have 6 copies of every chromosome. There'll be sort of pairs that are similar to each other. So there's kind of 3 genomes, each of them are diploid. And this is a huge challenge in terms of genomics. Firstly, in terms of scale, it's just a huge, huge genome. And secondly, in terms of repetitiveness, it means that everything is repeated 3 times. Makes it much harder to do things like alignments, much harder to assemble and create the genome. And then when you're actually taking it forward to do experiments with and to do things like knockouts or genome editing, every gene then has 6 copies. So you don't just, in something with only a diploid genome, you just have to make sure you edit it twice. With wheat, you have to edit a gene 6 times to make sure that your knockout or your edit is going to go through into the next generation and actually cause a phenotype. Everything that I've put a little E next to here can be found in Ensembl. So you can't. It's not just the wheat bread wheat genome itself that you can find in Ensembl, but most of the precursors as well are available genomes for study. So the genome that we have is the IWGSC RefSeq version 1.0. So this is 14.5 gigabases. It's made up of 21 chromosomes, which is 1 to 7 ABD. So it's chromosome 1 to 7, and then within each of the 3 genomes. And it does also include some unplaced scaffolds. The previous genome, if you do have data that's on the previous TGAC version 1 genome, you can access that via the October 2017 archive site, and this consists of unordered scaffolds assigned to chromosomes. So it's a slightly less assembled genome as the IWGSC genome. The annotation we have comes from the RefSeq version 1.1, and this has 2 different methods of automatic annotation combined with manual curation to produce 108,000 high confidence coding genes and a further 162,000 low confidence coding genes. And the gene names are assigned from the IWGSC. So we're going to have a look at finding out about a wheat gene. The gene we're going to look for is delta gliadin D1. So the D1 tells us it's on the D1 the D genome, and we're going to find its sequence, orthologues, homoeologues, and variants. And I've got just, this is sorry, the thing was bouncing at me. Is anybody going to jump in and can we deal with the questions while I carry on with the demo? We've got a question about high and low confidence genes. So I was going to look for this gene, delta gliadin D1. So I'm on the Ensembl Plants homepage at plants.ensembl.org. And there's lots of things that I can see here. So I can see which release number I'm in. We're in release 42, the Life, the Universe and Everything. We've got some top popular genomes that a lot of people access, with wheat being one of those. Wheat is actually our number 3 genome in all of Ensembl. So it's human, mouse, and then wheat in terms of popularity. So I'm going to use this search box to search for my gene of interest. And I just find a single gene. I click on the identifier, it takes me to the gene home page. So I've got the gene model here. I've got a table. It's only got 1 transcript, but it gives me a bit of information if I open up the table, telling me the size of the protein and things like that. And I've got a menu down the left here with loads of information of things that I might want to find out about my gene. So I might want to get its sequence. If I go into sequence, I've got the sequence here. So it's just got a single exon, which is highlighted here. I can change how this view looks, so it's just FASTA format. But if I go into configure this page, I can do things like show variants. So now we've got all of the genetic variants, which are highlighted on this sequence. I can click on them and I can find out a bit more about them. If I go into the transcript tab, I can also find other kinds of sequence, such as this cDNA sequence is really nice because it shows me the amino acid sequence along with the cDNA sequence. So that can be quite useful, particularly if you're interested in variation. If I go back to my gene tab, I'm going to have a look at more stuff to do with variation. I'm going to show you the variant table, which is here near the bottom of this list. And here I can see all of the variants which fall within this gene. And I can see whether they're missense, what amino acid change they have, all kinds of things like this. I can see orthologues. If I go into orthologues, I can see all the orthologues in different plants. So you can see this one actually has an orthologue in one of the wheat precursors and also in barley. And if I go into homoeologues, I can see we've got, so this is, if I look up here, this is my 1D version of the gene. I've got my 1B version, and I could do something like view the sequence alignment. So it's sort of 89 or 87% identical. I could see the protein alignment if I wanted between these two homoeologues. There is loads and loads to explore, but I'm not going to click through all of these different options on the left. It's just to give you a flavour of what's there. So I'm going to carry on and talk more about variation data. So I touched a little bit on variation data. So we have variation annotated. We have small scale variation and large scale variation. And we annotate variants in terms of the consequences they have on the genes that they fall in. So do they fall within coding regions? Do they fall within UTRs or splice sites or introns? If they do fall within coding regions, do they change the amino acid sequence? Are they missense? And I don't expect you to read this. This is just to point out that we do have a table listing all the consequences that we use. In Ensembl, we have them, they're ranked in order of how severe we consider them to be, and there's definitions of them, and they are standard sequence ontology terms. The variants we have for wheat, so we have variants from the axiom SNP array, and that includes allele frequencies from serials DB. We also have the EMS-induced tilling variants in Chronos and Cadenza wheat strains. And we've also called interhomoeologous variants between the different copies of the chromosomes. We use SIFT to predict the effect of missense variants. So SIFT scores changes in the amino acid sequence, and it's based on how well conserved the protein is and the chemical change in the amino acid. It's important to point out that SIFT predictions are always going to be predictions. They are never ever facts. They are not validated through experiment, and they will never be as good as experimental validation. So I'm going to search for a wheat variant, and we're going to find out about its frequency, its consequences, its frequency, and any sample genotypes, and we're going to find co-located variants. So I'm going to select the name of this variant. I'm going to go back to my Ensembl page, and I'm going to search for it. So this takes me to the variant page. So I've got another menu on the left with things that I can find out about the variant. I'm going to go into genes and regulation. And I can see this is the gene that it falls within. I could click through and find out more. It is, I can see the amino acid change, so it's a proline to a serine. I can see its position. And there's a SIFT score. It's listed as deleterious, but it's low confidence. Because SIFT depends on doing alignments between related proteins. If there aren't many related proteins that it can find conservation scores for, it will label something as low confidence, even though there may be only a little bit of, it may indicate that it's deleterious. It will give it as low confidence if it doesn't have many to compare to. So some other things we can take a look at. If I go into genotype frequency, you can see that according to the Axiom data set, so this, because this variant was taken from the Axiom group from Serials DB, we can see that the G genotype is the most frequent, the G allele and the GG genotype, whereas the AG genotype is a lot less frequent, and the AA genotype has not been observed by that group. If I go into the sample genotypes, I can get a bit more detail on this. And it will actually list for me all of the different samples and strains which had the different genotypes. So if I wanted to find a particular strain, if I was really interested in this, here we go. If I was interested in finding a plant which had this alternate allele at this variant, I could find it on this list and find a strain here. Another thing I could do is I could see if there's any cadenza data or chronos data. So here we've got a co-located variant from the ENS-induced mutation. If I click on this, you can see that it's also the same variant was introduced as part of the tilling populations, and you can buy the seeds from this from UK Seed Store. So if you found a variant that was really interesting to you, this could be a way that you would you could then find that and use that in your experiments. Another way that you might be approaching this, rather than having a known variant that you wanted to find out information about, it could be that you have your own variation data and you're looking to find out what information you can pull out of that data. So if you've got data from whole genome or whole exome sequencing, and you've created a VCF, you then want to determine what genes your variant hits, what consequences they have on those genes, and if the variants are already known in the database. And the way that you can do this is using the variant effect predictor, or VEP, and this is available as an online tool. It's available as an offline command line tool, and also as REST API endpoints. So what I'm going to demo today is using it in the online tool. The offline tool is all typing things in the command line, but the principle behind it is the same. So if you go into the materials section of the webinar interface, you'll see that there's a file there called wheat-VCF, which is what I'm going to use. So I've got this open here. This is what this VCF looks like. It's a very small VCF. It's about 100 variants long. So I'm just going to select it and copy. And I'm going to go back into Ensembl, and I'm going to go into Tools at the top of the page. I'm going to hit Variant Effect Predictor. And I'm going to run a new job. I need to tell it I'm working with wheat. There it is. And I'm going to paste in my data. And there's loads of extra information that I can get about it. I've got loads of options that I can open up here. So I can choose to get things like the gene symbol if it's available. I can get my output in HGVS. Unfortunately, the options in the VEP, they don't change depending on what species you're in. So they do give you a lot of human-specific options. you can get things like biotype of the transcript, protein domains. It will automatically get you SIFT predictions. And I can also filter if I wanted to only get variants in coding regions. If I now hit run, this is queued the data. It refreshes every 10 seconds. I can save the job to my account. The accounts are free and nobody ever sends you any spam emails. So if you want to set up an account and save jobs to it, you can do. I can also share the job. If I click on this little share icon here, what I will in fact do is share it to you all here. And I'm going to cheat while this is queued in case it decides to take a while, because I know that this job here underneath it is actually one that I did with exactly the same data the other day. So I'm just going to hit view results. So you can see it's put in 110 variants, gives me a summary. It's I didn't do any filters, so it hasn't filtered any of them out. 4 of those variants are novel, and 106 of them were already known in the database. I've overlapped 21 genes and 25 transcript. This is because it is a subset from a small region of the genome that I got this data from. And you can see I've got a table which lists the consequence, and it only shows us the first few variants because otherwise the table would get very, very long. But you can see what the allele is, what the consequence is, and on what gene that consequence occurs. And as I scroll across, I can see information about the existing variant. Since these are a couple of synonymous variants, at top and bottom, you'll see I've got codons, even though the amino acid is unchanged. I can do some filtering here, so I could say I want to see missense variants. It helps if I type it correctly. And it's having a bit of a think. I can filter by all kinds of things using these filters. So I've chosen this time to filter by consequence, but if I wanted to filter by something like seeing only novel variants or only variants that have been observed before, that's something I could filter by at this stage. You can filter before you run your query, but it is my preference to run the whole thing and do any filtering afterwards, and then you don't lose any data. Here we go. So we've got some missense variants. And as I scroll across the table, you'll see that they've got some SIFT scores, and these ones are actually these are showing in the green and red. So this one's deleterious and this one's tolerated, because we've got some more. There's more protein sequences that were used in order to create these scores, and we've got amino acids changes and things here. So I wasn't planning on it, but I think we've got time. I'm going to quickly go and have a look at Biomark. So Biomark can be really useful. The example I was given is if you've done a what's it called? If you've identified a haplotype, if you've been doing a breeding experiment and you've identified a haplotype through your breeding, which is associated with a beneficial phenotype, you may want to find all of the genes within that region of the haplotype in order to determine what might be causing this beneficial phenotype. And Biomark can be a great way to do this because it can generate lists of genes with bits of information about those genes, and you choose what that information is. So I'm going to choose my database. I'm going to choose genes. And I'm going to choose my wheat again. It just takes a moment once I've identified which data set it wants, it's going to take a moment to generate these options here. So what we call the filters and the attributes. So the filters represent which genes are actually going to end up in my table, and the attributes represent what information about those genes I'm going to print in my table. So if I go into my filters, and there's lots of things I can filter by, such as gene ontology or gene names or anything like that. I'm going to go into region, and I'm going to pick a slightly arbitrary region. It doesn't really matter what the region is. So I think that's quite a big region there. So I've picked my chromosome and scaffold, and I've picked my coordinates. I can combine filters as well. So if I wanted to find, say, all genes within this region that were associated with a particular function that I identified by gene ontology, perhaps because I knew what this phenotype was, and I thought, well, they're all going to be ones that are involved in this particular thing, then I could add an extra filter there. Of course, then you'll be missing anything that hasn't yet been annotated with that gene ontology, and your haplotype is going to be the thing that adds that information. So now I'm going to go into attributes and choose the things that I'm actually going to print into my table about these data. So by default, it's going to get the gene and transcript stable ID. And if I open up gene here, I'm going to add gene description. And if I expand up external, let's say I wanted to compare to other databases, I'm going to put NTBI gene ID, and I'm going to pick my Go terms as well, because I do want to find out about the functions of these genes if there is anything known. Now, if I hit results, it has a bit of a think, but it will show me a table very shortly. It's not being particularly responsive for me today. So here we go, we've got our gene and transcript stable IDs. Not all of them have got a gene description or an NTBI gene ID, but we have the Go term information here, and it's only showing me a preview table of 10 rows. This means that did take a few moments to load, but if I've done a query that's going to give me 100,000 rows, you can imagine that would have taken even longer. By giving me a 10-row preview, it means that if I now realize that I've missed a column that I needed to get extra data for or something like that, I only waited a little bit of time. It's very easy for me to go back. If I just spent 20 minutes waiting for 100,000 rows to appear and then discovered I'd made a mistake, then I would not be happy. I think I saw something about the filtering by the high confidence genes. I'm not sure, but let's take a look. I can't see anything that's a filter. No, I don't think so, but what we can do is we can request that. Another thing I might want to download, if I go into attributes, I can also get the sequences. So I could get the sequences, I could download, say, cDNA, and I could get the sequences if I needed that for taking forward to a further experiment. I can get homoeologues. There's all kinds of things that are available to me about my list of genes within my region of interest. There we go, there's my sequences. So yes, for large scale data, I've mentioned Biomar already. I've also mentioned briefly the FTP site. If you do want whole genome downloads, so if you wanted to do something like analyze RNA-seq data or something like that, you probably want to get flat files, which you can get from the FTP site. I'll also mention the REST API, which allows for language agnostic interaction with the database. There is an online course on EBI Train Online on using our REST API, which I do strongly recommend. There is loads of help and documentation on using Ensembl. I will apologise that most of the videos and things you'll see will be using human as their example, because it is our #1 species, and wheat is down at #3. But almost everything is applicable across onto what you might be doing with wheat. You can also follow us on social media, and you can have your own course. We can come and teach an ensemble course at your institute. The courses are free. We ask you to pay the trainer's expenses, but beyond that, we don't charge any fees. We can do browser courses, we can do REST API courses, we can also do train the trainer courses. Please send us an email. We are helpdesk@ensemble.org. We also have publications. If you use our data, please do cite us. And this was the team at our latest retreat. These are our funders, who we love because they fund us. And there are some upcoming webinars from the EBI that you may be interested in. Please take a look at the programme for the upcoming EBI webinars, and there's a survey that will launch after the webinar today. There's loads of stuff that could be potentially useful, and not just in the webinars. If you take a look at Train Online or the list of courses, there's loads and loads of stuff that's really potentially useful. The Train Online stuff can be done in your own time as well, which is really helpful. And there is an in-person training course which may be of value, which is Bioinformatics for plant biology, which is planned for November. It's a bit of a stub article at the moment. There isn't really information on the timetable, and you can't register. You can only just register interest, and then you'll get an email to tell you when it's opened up. But if you are interested in that, I only added this slide about just before we started this, so I'll put the link to that in the chat box, because it won't be in the slides that you can, or Melissa might do that, because it won't be in the slides that you can download. Are there any questions? Is there anything that you would like to know how to do with Ensembl Plants that I can demo on the screen? Right, somebody asked after the GFF 3 files. So if I go to the home page of Ensembl Plants, and I go into, there are a couple of ways to do this. One way is to go into your plant of interest, and then you can click on the link here to GFF3, and that will take you to the correct page in the FTP site. You don't need to use an FTP protocol. You can just click on the links and they will download. So if you don't have any kind of FTP protocol installed, you don't need to use it. Similarly, if I go to downloads, I get a list of all the species, and I can filter. And I've got the links here. Again, it will take you to the FTP site and give you the opportunity to download, but you can just download them. You don't need any kind of FTP protocol set up. Was there anything else that I ought to show on the screen? Okay, the difference between one-to-one homologue and one-to-many homologue. Right, let's find a gene. Let's see if I can go back to my gene. This gene wasn't very good because it didn't have that many homologues. So this has got, right, so here's a good, it's got a one-to-one orthologue. So what this means is that this species has one gene and wheat has one gene, and they're perfectly matched. If we compare it with Hordeum vulgare, with barley, it's a one-to-many, which means that a duplication has occurred in the lineage in barley. Barley's got 2 copies of this gene that are paralogous to each other, whereas wheat's only got 1 copy. So that's what we mean by one-to-one and one-to-many. We also have many-to-many, which would be when the duplication has occurred in both species, has occurred after speciation, in which case there are paralogues in both species, but there's no kind of direct one-to-one matching. There's a group here and there's a group here. Right, the wheat tilling dataset is very large and it's impossible to fetch it by chromosome or even by effect type, as I ran into timeout error. And yes, on our biomart, yes, biomart is not suitable for whole genome download in any way. Biomart cannot do that, particularly not with variation data. If we go back to the FTP site pages and we go back, then your best thing to do is download the VCF for the species and get all the variation rather than attempting to do it through Biomart. Biomart's not intended for that at all. Right, is there an option to design primers in Ensembl? We don't have a primer design tool. You can use our sequences, and certainly a lot of people do use our sequence views that we looked at in the gene tab and the transcript tab to then put the sequences into something like primer 3. Things like there was a page that I didn't show you, but I'll just, even though this gene isn't great for it, if I go into the transcript tab, there's this exons page. And so this shows the exons, and then that if it's got introns, which this one doesn't, it shows you the intron and the exon, it shows you the length of each of them. If you wanted to do something like RT-PCR primers, then this could be quite useful because you can see the length of the exons and the length of the introns and how long a fragment you're likely to get. So people do use this view for that sort of thing. Have we got any more questions? If not, thank you very much, everyone, for attending. I hope you use Ensembl Plants in future.